and she is an associate professor and in the internal medicine, and also associate director of the medical scientist training program in the Ohio State University. And I know him just a couple of years ago, and one day when I was the president of the OASOT, and we need to nominate some new board member, and someone suggested her name. And when I look for the online page, and I strongly support, and I nominate her and as our council and member. And she already served that for 3 years right now.
My third year,
yeah. And she did a good job. And from the CV I learned, and she was very systematic training and very interesting. She got like a bachelor in animal science and immunology, and for master in immunology and poultry science.
Yeah.
And for the Ph.D. and molecular biomedical science and immunology and toxicology. So that is very diverse. And she did a postdoc in the Dept and in 2011. And she got a second postdoc fellowship in the NIH and EHS. And in the research triangle, and that is in 2014. And then she recruited as an assistant professor by East Carolina University, and stayed there until 2000. And then she been recruited as an associate professor, and with tenure from the 2020 and in the current location. And Kim research is very strong, and she already building up in this place, and with a strong team work on the air pollution and information on the lung disease, and that is focused on the infection and the inflammation part. And she has been supported by 3R1 and also one U54, I don't know that, but as a PI, and she also have a one R21 right now is a 6.5% is pending. Is a
we'll see.
Good news?
Still haven't gotten it open, so we'll see.
And she also as a PI and the co-PI for several other foundation grants. So that is excellent. And based on the strong research, she already published more than 60 the research articles. And only in 2023 already published 9, so very impressive. And at one point, and for her first R1, and the percentile very high, 1 percentile. And she is active and a very good mentor, training for a lot of trainees. And she already been that for that and got an award from the Heart and the Lung Research Institute, Melissa Hyper Distinguished Mentor. And so that is excellent. And right now, she has some clinical trainee and also PhD trainee. And she is very active, involved in editor board as an associate editor for Drug Science. And she also is active, involved several study section and review grant. And she is a standing member right now is a SIEE, and that is related to the environmental science. And today she will talk about some the diet and the pulmonary response to environmental exposure. Yeah, what that, and she will introduce you.
Thank you so much, Lou. I really appreciate it. That was a very flattering. Hope I pulled up to that. But thank you all for the invitation to speak to you all. I'm very excited. This is my first time in Louisville. My husband has requested I stop and bring bourbon on the way home. But yeah, it's been an amazing since I visit so far. But yeah, so this talks to me more focused on the avenue of our lab that's basically looking at kind of how diet can affect your basically your responses to environmental exposures. But since I know that cardiovascular health is a huge emphasis here at University of Louisville, one of my goals today is actually to get you all to understand that the lungs do matter. They work very much in concert with the heart. And, you know, I mean, the lungs' most important function is gas exchange. We know this through our physiology courses that we've taken in graduate school or medical school. but also what we're breathing in every day is not clean. So you got to think the lung almost has to make a decision every time you breathe in something that might be noxious or infectious. And so to do this, we're obviously SARS-CoV-2 has really highlighted that the air we're breathing in has viruses in it. Also, particulate matter, we've seen that a lot with the wildfire exposures that we have been going through basically every summer for the last couple of years, and it's going to get worse. And then also allergens. Before moving to Ohio, I did not have allergies. I am now doing allergy shots. So thank you, Ragweed. Really appreciate that. As well as I'm a mom of an asthmatic child. So I'm very acutely aware of the allergens we breathe in. But then also microbial products. So endotoxin, et cetera. All of this is in the dust that we're breathing in on a daily basis. And then, of course, noxious gases, such as oxidant gases like ozone. And so when our lungs basically breathe in these different kind of stimuli, we basically first rely on the defense of the epithelial cells, which, obviously line, obviously the upper and the lower airways. And then, of course, this resident population of immune cells, the alveolar macrophages. And so once these different resident cells actually sense that these different stimuli are present, they are then, of course, going to set off a chemokine gradient, which is going to basically recruit into the innate immune response, which today we're going to mostly focus on neutrophils. And so these neutrophils are great. When I teach immunology, I kind of talked about them as like the big dumb bouncers of the immune response. So they're amazing at clearing out pathogens, but they spew a lot of reactive oxygen species and proteases that can actually cause a lot of damages. So, obviously you need to have kind of a tight hold on this neutrophil response. And so following this neutrophil response, what you're hopeful is that basically the adaptive immune response comes in and basically sets up memory. And so this is going to be mostly dendritic cells as well as lymphocytes, B and T. And so this memory response is great because it basically is going to set up the lungs so it remembers the next time it actually encounters these different environmental challenges, and hopefully you have a faster response so that way the lung is better prepared to handle them in the future. So I've told you this great story about these different immune cells and how they work to basically protect the lung from these environmental challenges. But don't forget that this immune response is beneficial, but it also can cause irreversible tissue damage. So too much of this immune response basically can be very damaging and obviously is going to impair the lung's own most important function, which is gas exchange. And so because the air we breathe in is not clean, these are numbers before the COVID-19 pandemic, but lung diseases were currently making up more than 15% of deaths worldwide. So we really believe that if we have a better handle on how the lungs immune response happens, especially to non-infectious stimuli, such as, particulate matter, oxidant gases, or even some of these allergens, we will be able to better target it through therapeutics. And so basically just kind of going over the central hypothesis of my lab, we mostly focus again on the impact of environmental exposures on the lung. And then also we're very interested in how this can influence pulmonary host defense response. So basically how you respond then to an infectious stimuli. But then through my my postdoc training at CIEHS, I became very acutely aware of the role of lipid metabolism in a lot of lung diseases. So what I've tried to do in my research program is basically combine these three arms and basically say that they work together and that, to better understand how the lung handles environmental exposures. And so the story I'm going to tell you at 4 p.m. today, I don't know where the talk is, but is mostly focused on iron, hemoglobin, and oxidant lung injuries. So if you're interested in any of that, please come and see me give a different talk at 4 p.m. But the one that you guys get to be, listen to this morning is going to be focused on basically the omega-3 DHA and lung inflammation. And so this is the state of global air. I stole it off their website. And basically it's showing you what people are dying from, which is basically down here. And then what can basically influence what they're dying from. And so that's right here. So we've got high blood pressure, smoking, blood glucose, BMI, etc. But in the top 10, you can definitely appreciate that we've got two things that are environmental: ambient particulate matter, household air pollution. And if you look at the green bars, which are basically, dying from chronic respiratory diseases, they really have a very heavy influence with air pollution on the incidence of people dying from chronic respiratory diseases. And even if you look down here at number 33, which is my favorite air pollutant, ozone, it has a direct effect on people dying from chronic respiratory diseases. So this association, I know I'm kind of preaching to the choir, has been known for decades, but we really don't know the biological mechanisms of how this interaction of environment and chronic respiratory disease, morbidity and mortality interact. And so what we do know, and this is a schematic, I have to say, has not changed much since I got my PhD. I'm not going to date myself, but it was a couple of years ago. So we do need more people to basically study how air pollution can induce pulmonary inflammation. And what we think happens, mind you, I'd say there's a bit of a black box here, is you inhale particulate matter, diesel exhaust particles, or even ozone. And we think it basically reacts with the lung lining fluid to basically create these damage-associated molecular patterns, or it can even carry in these pathogen-associated molecular patterns, such as endotoxin. And so these PAMPs and DAMPs are then generated in the lung, and they basically will then interact with innate immune receptors, such as toll-like receptors on the alveolar macrophage, and basically set off this kind of inflammatory signaling cascade that basically results in the macrophage producing pro-inflammatory cytokines and chemokines. These pro-inflammatory cytokines and chemokines then act on the airway epithelium, making it basically more leaky and allowing those neutrophils to basically come in and cause damage. So again, this is what we currently know, but we really need to add more to this schematic so we have a better understanding of, again, how something that's non-infectious like air pollution is basically triggering pulmonary inflammation. And so for this, we've been mostly focusing in my lab on ozone exposure. And I know that you all do a great job of studying air pollution. Specifically, I was going to say a lot of the criteria air pollutants. We've been mostly focusing on ozone, again, because it enhances respiratory and cardiovascular mortality, like I showed you in that state of the global air figure before. But then also because ozone levels are rising as ambient temperatures increase. And so, we know that an increase in ambient levels, such as an ozone action day, decreases lung function. You see more people go into the emergency room, mostly for cardiovascular or pulmonary complications. And even within 24 hours, you can see that hospital admissions are increased after that ozone action day. And so again, there's been some nice studies really tying in ozone and asthma and ozone and COPD, but the literature is really nicely showing us now that it's other respiratory diseases that it can exacerbate, such as idiopathic pulmonary fibrosis and even acute respiratory distress syndrome. There's been some nice epi studies that have shown that when you have an ozone action day, the severity of those chronic lung diseases are also increased. So definitely a pollutant that we need, again, more information on, specifically in the context of respiratory morbidity and mortality. And so one thing that has been known about ozone for decades is that ozone changes lipid metabolism. It actually can alter lipid metabolism not only in the lung. This was some work that was mostly pioneered by Bob Devlin's group at the EPA, where basically they showed that you're getting an increase in a lot of the pro-inflammatory eicosanoids, such as thromboxane, prostaglandins, and even leukotrienes in the lung, as well as systemically. And then if you looked actually even in like a non-human primate model, this has also been shown that these pro-inflammatory lipid mediators are upregulated, but then also even the enzymes that make them are actually increased, have increased expression in the lung after ozone exposure. And so the data that really got us excited was this really nice study that Irmela Codavanti at the Environmental Protection Agency did. And what they did was they actually did controlled human ozone exposures. And then they took blood from these patients and looked at different untargeted lipidomics. And basically they were seeing kind of these global alterations about 24 hours after controlled human ozone exposure in human plasma. More specifically, there was a lot more polyunsaturated fatty acids that were circulating in the blood of these patients after an ozone exposure, really indicating to us that there's a global alteration in lipid metabolism following exposure to ozone. And then they were also followed up with this with a really nice rat study where they were actually showing the same thing in the rat, not only in the blood, but also in the liver. They were able to see that there was an increase in circulating and pulmonary, not liver, polyunsaturated fatty acids. So this was really a kind of a nice bench to bedside study that really indicated that lipid metabolism was definitely something that ozone, is definitely altering and could potentially be some of the contributions to why ozone induces pulmonary inflammation. And so for those of you who study lipid metabolism, this will probably be a review, but just to bring the whole audience up to speed, we know, again, that when you encounter an injury or an infection, our body is really fast at producing these pro-inflammatory lipid mediators, such as prostaglandins and leukotrienes. And we have a lot of therapeutics on the market, especially with asthma, that actually can target these different pro-inflammatory lipid mediators to try to dampen their inflammatory response. But basically, the production of these pro-inflammatory lipid mediators drives inflammation. And so for a long time, we thought that this inflammatory response kind of just petered out, right? So it just kind of, waned. But Dr. Charlie Serhan as well actually was really able to pioneer over, I'd say, the last 15 to 20 years that no, it's not a actual, passive progress, that actually resolving this inflammatory response is more of an active process. And this is through the production of these different specialized pro-resolving lipid mediators, which are mostly, again, your lipoxins, your resolvins, your protectins, and even your mucins. And basically, production of these causes the body to basically resolve that inflammation. And if you don't have the production of these SPMs, you basically get more prostaglandins and leukotrienes being produced, and this can lead to chronic inflammation. So this is really it's called lipid class switching. And so you can really appreciate that lipid metabolism leads to both inflammation as well as the resolution of inflammation. And so how do these SPMs get made? Well, they're mostly made by N3 polyunsaturated fatty acids, so such as your eicosapentaenoic acid or docosahexaenoic acid, so EPA and DHA. But when also arachidonic acid can even make some of them, such as lipoxins. So basically through the 12-15-LOX enzyme, or even COX-2, or even your P450 enzymes, these an N3 or N6 PUFAs are then basically metabolized into these intermediates. And then 5-lipoxygenase basically creates the oxidation of these SPMs, and you can appreciate their chemical structure down here. So once these SPMs are made, what they're known to do to basically stop inflammation is they actually stop neutrophil transmigration and chemotaxis, so stopping that innate immune response. They also can block the production of prostaglandins and leukotrienes. And they do this, we think, by competing with 5-LOX for basically those enzymes. Again, they reduce pro-inflammatory cytokine release and even the function of those cytokines. And then also they're really important in getting non-inflammatory monocytes to come in and basically kind of clean up the tissue damage that has occurred. And then, they also are really, really important in promoting what's called macrophage efferocytosis. And this is basically macrophages eating dead cells. So therefore, you don't have necrosis, secondary necrosis that can lead to further inflammation. And then it's not just, dampening the immune response, but they also are really important in antimicrobial defense mechanisms. There's been a lot of really great studies with SPMs showing that, they not only dampen inflammation, but they're really good at helping you clear pathogens. So I think that that's kind of a cool duality that these lipid mediators have. And so how do they do these amazing functions? Well, they actually function through G protein coupled receptors. I'm only showing you 5 right here for the simplicity, but I think we're up to 9 different G protein coupled receptors that these SPMs can bind to. And so these G protein coupled receptors, downstream of them, you can get basically a decrease in COX-2 expression, which basically leads to less leukotriene production. They actually can block NF-kappa B, so then you don't have that host of pro-inflammatory cytokines and chemokines being produced. They are known to actually increase IL-10 production and even increase antioxidant response elements such as heme oxygenase 1. And so again, just basically these are the receptors up here. And, they do seem to bind some specific specificity. So like toxins and resolvents bind to ALX FPR2, resolving E's, which are downstream of EPA, bind ChemR23 or ERV. And then even some of these RVEs can also bind BLT1. And we've actually made knockout mice for both this receptor and this receptor, if anyone's interested in pursuing those in your research. And so SPMs and lung disease, we have seen some data. It's been mostly out of Bruce Levy's lab at the Brigham, showing that there is an association with SPMs and chronic lung diseases. They've been mostly pioneering this in the context of allergic airway responsiveness, where basically if they treat mice with these SPMs, they're able to see that with an allergen challenge, they don't see a lot of inflammation, as well as they see less airway hyperresponsiveness. Acute lung injury, there's also been a group that has shown that basically with acid aspiration, if the mice have an adequate amount of SPMs, there's reduced mucosal inflammation. And if there's a deficiency in SPMs, there's more mucosal inflammation. And then bacterial pneumonia, like I said previously, there's been some gram-negative and gram-positive studies that have shown that basically mice that are infected with these pathogens, if they give them SPMs, they actually clear the pathogen faster, and the tissue restores to homeostasis, also at an accelerated timeline. But around the time we were studying these studies, there was only one group, and that was Patricia Syme's group at VCU, that was looking at SPMs in the context of something that was, kind of environmental in focus. And they were doing it in the context of cigarette smoke leading to COPD. And one thing they were able to show actually through not only COPD patients, but then also in a mouse model of cigarette smoking, is that the macrophage phenotype was basically changed with SPMs. And that the macrophages had more of kind of an anti-inflammatory phenotype, as well as they ate more apoptotic cells if they were cultured with SPMs. And basically also they were able to see that there was a resolution of lung inflammation following a chronic cigarette smoke exposure in their mouse models. So this kind of really told us that, maybe this is a good target for something more environmental, and maybe SPMs might be critical in the context of ozone exposure. So just to kind of recap what I told you a little bit for our current knowledge gaps in air pollution research before I go into our data. Again, pulmonary exposure to these air pollutants, including ozone, leads to inflammation. And that is basically characterized where we see more, we have inflammation in the lung as well as systemic inflammation. And then also you see an increase in reactive oxygen species, as well as those pro-inflammatory lipid mediators that I described earlier. And then also, lung permeability is increased. So the lung, again, is more leaky, but how this happens, we really don't know. But we were hypothesizing that maybe this is due to an alteration in SPM production and or signaling. And so to do this, it was actually my first postdoc, Dr. Britta Kilberg-Bassnett, who's now actually at Covance in Madison doing very well. But basically what I tasked her to do was, let's just do, an acute ozone exposure that basically replicates what you would see on an ozone action day. And let's do lipidomics and see if we can see some changes in the SPMs. And so to do this, basically, she took C57 Black 6J male mice. We have also done this with females and exposed them into either filtered air or 1 part per billion of ozone for 3 hours. So 1 part per million of ozone kind of is equivalent to about 200 parts per billion for humans. So very much what you would see on an ozone action day. And again, that 3-hour time point is also more like what we're exposed to on an ozone action day. And then 24 hours post-exposure, this is usually when we see that inflammatory response to ozone. We're going to take bronchial alveolar lavage fluid, we're going to take blood, and we're also going to take lung and spleen tissue for non-targeted lipidomics. And so the data she got back from this study, obviously prostaglandins, leukotrienes, those pro-inflammatory lipid mediators were up, but she was really having to dig to see if she could see any differences in some of the SPMs we were able to detect. And, just to have you appreciate what this inflammatory response looks like 24-hour post-ozone. So again, I'm not sure how many people in here have done some ozone exposures, but what you're looking at is not a sledgehammer inducing inflammation. It's kind of this low smoldering inflammatory response. And this is looking at the BAL cell differentials in the lungs. So you can really appreciate that there's an increase in macrophages in the airspace, as well as about a 5% increase in neutrophils. And then if you look at markers of lung injury, such as total BAL protein or BAL albumin, you can really appreciate that the ozone-exposed mice, which is this black bar right here, have a significant increase in the amount of leakiness that their lungs currently have. So really kind of setting the stage that we do have an active inflammatory response to this dose of ozone. And so again, what she was seeing, again, when she got this lipidomics data back was more, your prototypical pro-inflammatory lipid mediators were upregulated. But when she was looking at the SPMs, she wasn't seeing across the board this different change. If anything, what she was seeing is that it was specific to the SPMs that were downstream of DHA or docosanoic acid. I always screw that up, I'm sorry. And so just to kind of review with you what that pathway looks like. So basically, if you have DHA in your cells through either 12-15 LOX or COX-2, you basically can get the production of 17-HDHA or 14-HDHA. And then 17-HDHA is further metabolized by 5-LOX into the resolving D series, whereas 14-HDHA can be its own signaling molecule, so it's a monohydroxy DHA, or it can be further, oxidized into myrcene 1. And so this was really the pathway where she started seeing hits, which I thought was very interesting. It's like, why specifically is, the ozone targeting kind of these downstream DHA derived SPMs? And so these are the data we actually published back in 2018, but just to kind of go over it to set the stage, you can really appreciate that what we saw during an active inflammatory response wasn't that the SPMs were upregulated, which is kind of what we thought. You got more inflammation, you would think you're making more SPMs. If anything, what we were seeing is that the SPM production in the lung was suppressed if it was downstream of DHA. So you can appreciate that. Again, the ozone-exposed mice had less 14-HDHA, again, which is a monohydroxy DHA molecule that can do some of its own signaling or can be produced or can make myrcene 1. 17-HDHA, and even protectin DX. So those three were the ones she really saw reductions in. And this wasn't just in the lung, much like the Codavanti group saw. We were seeing that this was also in the spleen, really indicating that there was kind of this global alteration in lipid metabolism following ozone exposure. But it did seem to be downstream of this specific N3 PUFA. And so then just to, understand why it was happening, we went on to actually measure 15 LOX and 5 LOX expression in the lung tissue following ozone exposure. And again, much like we were seeing with the SPMs, 12-15 LOX was significantly decreased in the lung tissue following ozone exposure, really kind of indicating that, here is where kind of the enzymes might be, basically unable to produce these SPMs. And then asking, okay, you've got decreased SPMs. What's this actually doing to the signaling? So to do this, we did real-time PCR for specific SPM receptors. So BLT1 and ChemR23, again, bind your resolving E's, whereas ALX FPR2 actually bind lipoxin A4 as well as the resolving D families. And so what we saw actually, again, very much mimicked what the DHA-derived SPM precursors and SPMs showed us, that, where we had less of these SPMs being produced, the receptor expression in the lung tissue was also altered, really kind of indicating to us that, yes, we're getting kind of, less production that's really kind of shutting down some of the signaling pathway. And we've got more detail in this paper if anyone's interested in. So, but then I kind of tasked her. I was like, okay, you're seeing this cocktail of DHA-derived SPM precursors or SPMs. What if you gave it back to those mice? What does that actually do to the inflammatory response? So what she did was an IP injection of these 3 DHA-derived SPMs before ozone exposure. And then again, went through our typical filtered air or 1 part per million ozone exposure regimen that I described earlier. And again, we're taking, again, tissues and BAL and blood 24 hours post-exposure, basically to understand if replacing these SPMs did anything to the lung injury and inflammation that we see following ozone exposure. And so this is just to give you an idea that the orange bars are basically the ones that got the pre-treatment with that SPM cocktail. But basically, we're giving an IP and it does reach the lung. We have tried oropharyngeal aspiration. We don't get the same results with oropharyngeal aspiration, but I think that's also because ozone probably blows up a lot of those lipids. But I do think that this is basically really indicating to us that, there's a systemic production of a lot of these omega-3 derived SPMs that then can affect the lung. But basically giving them IP, we are seeing that there's an enrichment of these SPMs in the lung 24 hours following exposure. And so then to look at the inflammatory response, sorry, I know this is a bit busy, but these are our VAL cell differentials. You can basically appreciate that pre-treatment with these SPMs really does lower the macrophage numbers in the airspace following ozone. And then also we're seeing kind of a decrease in the neutrophils that are recruited into the lung following ozone exposure. I will point out that this is not like a complete abolishment of the neutrophil response. There's still a small amount of neutrophilia, which I think is really interesting because maybe we need that small amount to basically promote resolution of tissue inflammation. But where it gets a little complicated is when we look at markers of lung injury, and I'm only showing you BAL protein. This cocktail of SPMs pre-treatment really affected the inflammatory response. It didn't do anything to any markers of lung injury. So I really think that this tells us two things. One, that inflammation and injury are probably driven by very different pathways in the lung following ozone exposure. But also, maybe this is specific to a systemic response that these SPMs actually have that basically can affect the amount of cells that are infiltrating the lung following ozone exposure, but it doesn't do anything to kind of barrier permeability mechanisms. So still trying to wrap our heads around that. But then when we looked at kind of cytokines and chemokines, it was not across the board that we were seeing that this SPM cocktail really was having an effect. If anything, it was very selective. And so we were really seeing a difference in this pre-treatment with the SPM cocktail before ozone exposure lowered IL-6 production, as well as IL-1 beta, and then only one of the neutrophil chemokines, which is CXCL1. So this really to us indicated that there might be some selectivity in how these SPMs act, especially in our ozone model. It's not across the board anti-inflammatory, seeing as some of our mediators were unchanged. And so then, of course, the hallmark of tissue resolution is macrophage efferocytosis. And again, as I told you before, basically macrophages eat dead cells. And when they eat these dead cells, they basically produce a lot of anti-inflammatory mediators, and this really promotes tissue homeostasis. There's been a lot of different cardiovascular studies as well as pulmonary studies really showing that if this process is, kind of messed up, you basically get this chronic inflammatory response. And so one thing I also think is kind of cool, besides the fact that, when a macrophage eats a dead cell, it produces these anti-inflammatory mediators, but it also kind of like talks to the other macrophages around it and really promotes them to do this efferocytic capability as well. So it's kind of like, they all follow the leader. So really, if one's messed up, a lot of them are going to be messed up. But this was really, in my mind, our way to see if that SPM cocktail was actually, altering resolution of inflammation, was to show that we had changes in this efferocytic capability. And again, impaired efferocytic capability of macrophages has been linked to pulmonary disease progression. So definitely would kind of indicate to us why air pollution might promote a lot of chronic inflammatory diseases is through maybe, alterations in this macrophage efferocytic function. And so this is my former grad student, Miles Hodge, who actually now is at the EPA. I basically texted him. His whole entire PhD was focused on basically how, ozone can impair pulmonary efferocytosis. So what we're doing, again, is exposing our mice to filtered air or ozone, and then 24 hours post-exposure, we're going to basically give these mice an oropharyngeal aspiration of apoptotic thymocytes. So we're basically feeding those macrophages dead cells. And then you basically can either come up with a phagocytic index where you can see that, by light microscopes that, the macrophage has this nice, purple dot, which is basically indicating it's taken up one of these thymocytes, or we even have a flow cytometry based assay that basically gives us, again, the amount of macrophages and how many have had eaten dead cells. And so basically what he came up with And this is the 24-hour time point. What I'm showing you is that ozone does impair efferocytosis in alveolar macrophages. So 24 hours post-exposure, we're getting about a 50% reduction in the macrophage efferocytic capability. And so I'm only showing you the 24-hour data for right now, but we do have this data in his publication out to 72 hours post-exposure. So these macrophages are really kind of stunned. So they're really not able to kind of, eat those dead cells and therefore promote resolution after this one hit of ozone. And we're currently trying to follow that up by doing some RNA-seq on the alveolar macrophages to understand, basically what pathways might be impaired. But then I asked him, what happens if you gave our SPM cocktail beforehand? What does that do to the macrophage efferocytic function following ozone? And sure enough, it restores it. So you can see here that ozone really knocks it down again. SPM treatment alone doesn't seem to enhance efferocytic capabilities. I really do think you need that inflammatory cascade to kind of promote efferocytosis. But then you can definitely appreciate that if the mice were exposed to ozone but pre-treated with the SPMs, we get kind of a rescue of this efferocytic capability. So, really indicating that, the macrophages are able to function better if they've been given this SPM cocktail. And so just kind of interim summary before I go into the diet aspect of the talk, ozone, I hope I've convinced you, ozone induces pulmonary inflammation while decreasing SPMs that are DHA derived, as well as some of the SPM receptors. And that SPM supplementation of the ones that were suppressed following ozone does seem to improve pulmonary inflammation in lungs and downregulate expression of select cytokines and chemokines. And then, of course, the macrophage efferocytic capability that I just went through, this pretreatment with SPMs really does seem to rescue the ozone-induced changes in the macrophage function. So around the time that I was wrapping up these studies, I had the pleasure, I was at East Carolina University working with a lipid biochemist, Raj Sheikh. And I joke that he's like the brother I never wanted, but we get along really well. We're very frank with each other. So I'm showing him this data, and he does a lot with like kind of obesity and nutrition and lipid medicine. And he was like, so what? You gave it back, so what? And he's like, what kind of human relevance does that have? He was like, because you're not going to give people IP injections of SPMs before a bad air quality day. And I was like, yeah, true point. And he was like, but it's a direct, basically reflection on DHA. So why aren't you just putting DHA in the diet? And I was like, I never thought of that. So we kind of worked together again, because he has a huge nutrition background, to start looking at diet and dietary PUFA intake. And how this might affect the ozone-induced inflammation. But just a little bit about diet, and especially dietary PUFAs. I think we've known for years that diet can influence cardiovascular disease, but who'd have thought that it might also have an indication on lung diseases? But there's been some really nice epi data that has actually shown that it can. And this has been mostly out of Nadia Hansel's group up at Hopkins, where they actually showed that kids that basically had an excess N6 PUFA or a Western diet, they actually, if they were asthmatic, had increased respiratory symptoms and lower lung function on bad air quality days. So they have this really nice, Baltimore kids cohort that they follow, and they were able to show that, yes, these kids do worse on bad air quality days if their diet was more representative of a Western diet, so high sugar and high saturated fat intake. And then, we've known for years that, DHA is an N3 PUFA that is poorly consumed in the Western diet. We don't eat very well. I think we all can appreciate that. And that currently, according to nutritionists, a significant portion of the United States is hypothesized to be deficient in DHA. And this is not only because of our poor diet, but there are also some SNPs in some of the enzymes that are important in kind of, your FADs and your elongation enzymes that basically allow us to incorporate DHA into our cells that are known to be in the population. So it not only might be diet, but it also might be genetics associated with this. And then, if you look at the data, especially, the human data, dietary DHA intake is associated with, some reduced inflammation, some it doesn't do anything. It's kind of all over the place. But I really do think that a lot of that has to come down to the quality of the supplements that are used in these clinical trials because, we as a country, unfortunately, regulate nutraceuticals poorly. And a lot of these fish oil supplements that are given in these clinical trials might have contaminating heavy metals. There might be oxidation upon generating these products, etc. But luckily for us, there is a product on the market, Vespasa, which has been shown very nicely to lower triglycerides. And this is kind of a purified N3-PUFA, more an EPA supplement, but it has shown some benefits, at least in the context of triglycerides. So it might be worthwhile seeing if, a higher purity, treatment has more cleaner efficacies in our different epidemiological studies. So again, Roz posed the question to me and we decided to take off and run with it. That if an SPM precursor or an SPM IP injection decreases ozone-induced inflammation, does dietary supplementation with the parent fatty acid, do the same? And so again, this is something that, I know nothing about diet, I will admit it, but I'm going to go through at least how we designed this study with Roz and hopefully answer any questions that might come up. But what we're trying to model here is basically, we have a purified lean chow and a purified lean chow plus DHA. I'll go through the components of the diet in the next slide. We start feeding these mice at 4 weeks of age, so right after weaning, and we're feeding them for 6 weeks. And the reason why we're doing this is we're basically trying to mimic growing up eating a DHA-enriched diet. Because I'm sure we all can appreciate getting an adult to change their diet is hard, and a lot of times we don't stick with it. So, and I'm guilty of that. But basically, if you grow up eating a certain way into adulthood, that might actually affect your responses. And so then after, the 6 weeks of feeding, we're going to expose these mice to either filtered air or 1 part per million of ozone. So that acute ambient increase in ozone. And then kind of do the same endpoints as I described to you earlier at 24 hours post-exposure. We do have some 48-hour data here that I'll go through as well. And so just remember that 24 hours is kind of peak inflammatory response. 48 hours is when the resolution response is about to start. And so the diet composition, because Roz tells me I have to clarify this to everyone every time I give this talk. So when I say normal chow, I mean purified lean controlled diet. So the only thing that is different in the DHA supplemented diet is DHA. So this is, again, the purified lean control diet plus 2% kilocalorie for DHA. We are using a very high purity of DHA. It's more than 93% pure. You would not believe the amount of money I pay for this purity of DHA. It's pretty insane. But the 2% kilocalorie is basically supposed to mimic a 170-pound adult eating about 2 grams a day. So very much what you would get in a supplement dose. It'd be a little hard to get up to 2 grams a day just by eating tons of fish, unless you live over in Iceland and have a primarily oily fish diet. We change this diet every 48 hours because, again, we're adding a lipid to the diet and it can oxidize very quickly, and we don't want to make these mice sick. So again, we are changing it quite frequently, and it's stored at -80. And then again, we're feeding for 6 weeks. So all these things, if you work with a nutritionist, are very important in your mouse studies, just in case anyone is interested in going into this avenue of research. So, and this is data again that my current PhD student, Michael Yeager, is currently working on. And I was going to say, and one thing that he showed, which I think is very, interesting, is that by the DHA supplementation in the diet, so this is the blue bars, we're really not seeing across the board that DHA in the diet really increases all DHA-derived mediators in the lung. And we have the same data for the spleen. It really seems to impact, again, this monohydroxylated 14-HDHA, which again can be, signal on its own, be its own kind of SPM, or it can actually be further metabolized into mucins, which are specifically made by macrophages and monocytes. So really understanding that we are basically enriching, this specific N3-PUFA in the lung, but I think it's specifically going into macrophages, and that's something he's currently trying to follow up on. But one thing I want you to appreciate is that, again, it really seems to be targeting this monohydroxylated DHA molecule, and that it basically does kind of rescue some of the ozone-derived decrements in the lung. And so what does this do to the injury response? So again, here we're looking at 24 hours post-exposure is kind of that peak inflammatory response, whereas 48 hours post-exposure, we're really looking at the kind of beginning of the resolution response. And so you can appreciate that, you know, again, ozone induces injury 24 hours post-exposure, and it doesn't matter if they're on the controlled diet or the DHA-derived diet, but we are seeing that the lungs do seem to resolve this injury response quicker if the mice had been fed that DHA supplemented diet. And then so looking at the BAL cell differentials, again, the data we saw was actually pretty comparable to that IP injection of that SPM cocktail. So we really are seeing that, you know, ozone exposed DHA fed mice have less macrophages in their airspace, as well as less neutrophilia when compared to the controlled diet fed mice. So indicating again that we're really kind of affecting that inflammatory response during active inflammation, but then the injury and inflammatory response basically are also decreased in the resolution and time points. And so then, you know, we run a whole host of cytokines and chemokines, which again, you know, there does seem to be, again, a selectivity in what DHA, can affect. But one that got us really excited is CCL2. So this is a monocyte chemoattractant protein. So basically gets monocytes into the lung, then they turn into macrophages. And so every single time we run this study, DHA in the diet really does knock down the expression as well as the production of this specific chemokine by more than 50%. So we're really seeing that it, you know, has a lot of selectivity. And so this was exciting to us because Deb Laskin's group at Rutgers has shown that the CCL2 or CCR2 deficient mice are actually protected from the ozone exposure. So indicating that really this kind of macrophage recruitment really does drive the inflammatory response, as well as CCL2 production following ozone exposure is very much dependent on a pattern recognition receptor called toll-like receptor 4. And so this really kind of gives us a nice pathway to chase down, which Michael is currently working on to see if DHA specifically can actually alter toll-4 signaling, leading to reduced CCL2. But again, to look at our ephrocytic function, much like the IP injection with the SPM precursor, DHA in the diet does seem to be actually rescuing the ozone-induced decrements in macrophage ephrocytic capability, which I think is why we're seeing that kind of, you know, lowering of some of the injury and inflammatory responses at the resolution time points that I mentioned before. And so around the time, again, I talked to Roz. Every time I talk to him, it's like, I don't know, I feel like I'm on Jeopardy to get asked all these questions. And so he was like, okay, you're going to go chase down this toll-4 pathway. You know, why aren't you looking at the lipid membrane? Because let me tell you, he's like, that's where all of these fatty acids end up. They're incorporated into phospholipids and then basically in the cell membrane. And so, you know, maybe all of your ephrocytic capabilities, et cetera, are basically changes in the biophysical movement of the membrane. And I was like, oh, great, here we go. I'm going down another avenue I know nothing about. But if I'm going to look at toll-like receptor 4, he is correct. You know, it depends on lipid rafts to basically induce their structure. And for those of you who are not familiar with lipid rafts, basically what they are is they're kind of this tight, compacted, lipid-rich area in the cell membrane where receptors go to signal. And so basically, if you have lipid rafts, you know, that are dysfunctional, less signaling will occur. And so sure enough, what we did was we actually, cultured MHS cells, which is basically a mouse macrophage cell line with DHA in the media or a media that was actually DHA deficient. And then we stained those membranes for choleratoxin B, which is basically a lipid raft marker. And what we saw again was a little confusing, but we're working through it. So if anything, DHA seems to promote lipid raft formation, which is strange because we're seeing that kind of those toll-4 signaling or CCL2 signaling is downregulated. So you would think that if anything, you would have less lipid rafts, but if anything, we seem to be having the promotion of rafts. So again, you know, Roz was great in helping us quantify the fluorophore area of this choleratoxin B staining. And then again, in typical lipid biochemist, way of thinking, he was like, let's take the cell out of it. Let's build some biomimetic membranes with DHA and see if that gives you the same perspective of lipid raft organizations. So I sent Michael to UNC to learn how to build biomimetic membranes, and sure enough, we got the same results that we did with the NHS cells, that if anything, what we're seeing is we're getting a promotion of lipid rafts in the biomimetic membranes that had DHA as a building block. So what we think is happening, and mind you, we're doing some membrane fluidity assays now, is we think that DHA is actually poking holes in the lipid raft, and so making it kind of bigger but less stable. And so therefore, if you think of it as like, you've got, holes in your raft, it's less likely that these receptors will be able to interact and actually create some of this downstream signaling. So that's what we're currently chasing, but again, unpublished data, so very open to feedback. And if you all think I'm crazy, I'm open to hearing about that as well. So, but I'll blame Ross. But yeah, so this is kind of the working model of what we think dietary DHA is doing in the lung. So again, we think that it's kind of promoting specific SPMs, more specifically that monohydroxylated 14-HDHA, perhaps through the production of myrcene or perhaps on its own. It's something we're teasing out. And then, might be binding to an SPM receptor that basically blocks NF-kappa B. We do have reductions in NF-kappa B in the alveolar macrophages, which is data I didn't have time to show you. But then another aspect is we really think it's disrupting this raft clustering in the membrane. So really kind of remodeling that cell membrane, which then can maybe affect some of these innate immune receptors and their signaling pathway, which ultimately leads to a reduced inflammatory response. But this is, again, what Michael's working on for his PhD, and I'm excited to see where this data takes us. But in summary, hopefully everyone's going to have salmon for lunch, but or take a soil supplement. I hope I've not only convinced you that the lung is, very worthwhile in the context of air pollution. Not to say the heart isn't, but they work together. But ozone does seem to be reducing select SPM levels in the lung while increasing inflammation. And that, both SPM supplementation and dietary DHA do seem to increase these pulmonary SPM levels. But then you also see a mitigation of the inflammatory response, specifically by less, immune cells in the airways, as well as kind of a selectivity of IL-6 and some in CCL2, perhaps through the TOL4 pathway. And then also this is having some effects on the cell membrane. So I really think that, unfortunately, biophysics is where we're going with a lot of this, but hopefully it will be some exciting learning curves. But overall, I really think that this is opening up this really cool new avenue for us that really highlights that, dietary fatty acids are going to have an effect on the lung. And so it might actually really impact our inflammatory response to not only, air pollution, but, different other environmental exposures. But maybe diet is also a way that we can kind of mitigate some of these global effects we're seeing. I'm sure nutritionists would argue with me on that, but, pie in the sky. And so these are some of the future directions we're working on. Again, we've got the heme iron story that I'm saying at 4. I have done a little bit of vascular function. It is improved with DHA in the diet, but I couldn't do a myograph by myself. I rely heavily on the great cardiovascular collaborators that got to do that. So, but if anyone is interested in not only some of the host event stuff we're working on, please come see my talk at 4. And then this is my group. Again, the majority of this data was done by, Michael Yeager, who's right there. He's about a year out from graduating. If anyone's looking for an amazing postdoc and somebody who's really in depth in macrophage biology and lipid metabolism, he'd be a great find. So, and that's, again, our lab website. And I guess it's not Twitter anymore. I guess you can follow me on, what is it, X now? Or I'm not sure. So, but thank you all for your attention. I'm happy to answer any questions.
Yeah. Very, very nice. I just want to follow up on the idea that DHA regulates the macrophage phenotype. So M1, M2, I didn't really see if that kind of fits into your.
I'm an immunologist, so I don't believe in the M1 versus M2, but Yeah, no, so we can take the macrophages out and I would say we didn't look for like your traditional IL-4, but I can say that a lot of pro-inflammatory cytokines are suppressed if DHA is in the diet. So as well as NF-kappa B is. But yeah, you're right. We haven't looked at a lot of those traditional kind of anti-inflammatory. We have looked at IL-10. We didn't see much of a change in IL-10, but I'm not sure if ozone is a great trigger of IL-10 too. So maybe it's even looking at something that might polarize them one way or another might be the way to go to see if DHA actually alters that. So. Yes.
I was wondering if you have any data on how ozone and or DHA is affecting the routing of low layer receptors onto the membrane.
Yeah, so intracellular versus extracellular. Yeah, so we haven't looked intracellular. What we've done so far is just the choletoxin B staining with TOL4. So whether or not that actually changes the trafficking, I'm not sure. And it's a great point because, you know, a lot of that intracellular trafficking is dependent on, you know, I was going to say those little endocytic vesicles, which obviously their membrane can be, you know, altered. I know Andre Holian at Montana has done a bunch of work with silica and basically showing, I think, no, sorry, it wasn't Andre Holian. It was James Pesca at Michigan State has done some work with silica and basically the internalization of silica and the breakdown of it in those little endocytic vesicles with DHA in the context of lupus. And they show that actually the silica is really good at kind of poking holes in those endocytic vesicles. That DHA makes the vesicle kind of stronger. And so therefore we have less silica that, you know, can trigger the inflammasome. So, but we haven't done it yet, unfortunately, but that's a great point that I'll probably bring up to Michael when I get back. Thank you. Alex, or if you, sorry. Yes,
I see you didn't really share any of it, but have you done any longer term exposure studies, repeated exposures?
We've done one of those 10-day exposures. It's like 10 days at 0.5 part per million that I think Jack Harkema is kind of, and we didn't do it with DHA, but we did it with the SPMs, and it does actually increase them by the 10-day. But I guess like I'm not set up to do chronic. I wish I was, like, because you're right, that's where we kind of need to go. But I feel like the 10-day, I wasn't sure what to make of it, you know, because you also have that eosinophilia that turns on 10 days post ozone. So yeah, it kind of reverses the more ozone goes, but I feel like the immune response also is very different at that time point too. So, but great point.
Yeah, very nice work. So I just have one question. Did you check the blood lipidomic change? And what kind of cells involved in this lipidomic operation in this specifically in the lung tissue or indirectly comes from other tissue, heart and liver?
Yeah, so we have done blood more specifically. We use the packed red blood cells to kind of target the phospholipid turnover. And so we can show actually that at least in the red blood cells that there's an enrichment in DHA with the diet. So it's kind of like a way we check. And then for the blood lipidomics, we actually didn't do blood, we did spleen. And that was me trying to, again, get at the immune cells. And so a lot of what we see in the lung is also in the spleen, but I really think that speaks to the fact that the immune cells are probably changing because the spleen is where they kind of home, right, to kind of talk to each other. So, but we haven't done the liver yet. I was going to say we've stored liver, especially after the DHA, because there is that long liver access, but we haven't done anything with it. But yeah, I'm very curious to see if like the liver perspective, at least in the lipid metabolism, is changed. And I know Irmela's group did see, you know, changes in polyunsaturated fatty acid levels in the blood in the human stuff. And I thought they did liver too, but I could be wrong. But they never looked at any of the SPMs or the DHA derived mediators. So I think that would be very interesting to understand if it's the whole body or if it's just the immune cells, you know, in the spleen and in the lung. So great point. Am I supposed to be looking for questions on Zoom?
I'll let you know if there's them.
Okay.
I guess the way I would think about them is that the lipid graphs are domains where the receptors go to become concentrated so
Yeah.
they can signal more.
Yep.
But if you have more lipid wrap and no increase in receptor number, then essentially it's not concentrated.
Right.
So is that, am I misunderstanding your idea about coating both? Is that what you're saying with coating the both?
Right, yeah, so even if they're there, I think that the poking of the holes doesn't allow the two receptors to talk to each other. So TOL4 needs CD14 to do a lot of the signaling. And cholera-toxin B actually is basically usually correlates with CD14 expression. And CD14 is constant, but TOL4 can kind of come in and out. So yeah, so that's our thought, is that basically that, you know, the number might not matter, but if they're not having that stable signaling platform, they're just not going to talk to each other. So we're doing, I think it's FRET, and I forget what else on, it becomes very complicated because again, Roz is not an immunologist. So we basically have to almost like fix cells and stain it and then send it to him for a lot of the physical movements. So I have a feeling I'll be taking a sabbatical at UNC at one point or another. So I can just harvest macrophages and give to him. So. No? Okay, thank you all.